Gonna go ahead and get started. So today we're very excited to have you all here and to share a little bit about our research in the synthesis, biological evaluation, and structure-activity relationship with diversified C4 analogs of podophyllotoxin as tubulin inhibitors. And there's a lot of big and long words on this title, but don't worry, we'll break it down. As Dr. Du mentioned, my name is Shreya, and I'm a senior at Lynbrook High School.
And I'm Stella, and I'm a senior at Harker.
So we'll start off our presentation with a quick introduction about the role of natural products and more specifically about podophyllotoxin. We'll then share some of the prior work done by our lab on C4 esters of podophyllotoxin, including the initial SAR hypothesis and some types of evaluation. We'll also include much of the work that we've currently been doing on the C4 carbonates and carbamates of podophyllotoxin, including their synthesis, biological evaluation, and a few conclusions. Finally, we'll cap today off with some future plans and different directions that our project may go
in. Okay, so podophyllotoxin is a natural product derived from a plant called the wild mandrake or Podophyllum pileatum. And for any Harry Potter fans out there, the wild mandrake is in fact the same plant as the magical screaming mandrake found in the Harry Potter movies. But moving on, the reason why our lab focuses so much on natural products is because, according to Newman and Craig in 2020, derivatives of natural products outnumber natural products themselves in a 10-to-1 ratio. And this just goes to show the importance of not only using, but also modifying natural products for medicinal purposes. Okay, so here's a visualization of podophyllotoxin's biological mechanism of action. The molecule binds to a protein called tubulin, and tubulin is responsible for the formation of spindle fibers, which are needed for cellular division or mitosis. So by binding to tubulin, podophyllotoxin is able to inhibit tubulin polymerization and thus prevent the spread of cancer. It's not switching slides for me.
That's so odd. Okay.
So a lot of researchers have synthesized and evaluated a bunch of podophyllotoxin derivatives, and they found that the C4 hydroxyl regulates the compound's mechanism of biological action. So in this slide, we wanted to highlight two compounds specifically, etoposide and teniposide. So these are two FDA-approved drugs, and they have these sugar groups at the C4 position rather than the hydroxyl group that podophyllotoxin does. And this slight modification at that one C4 position completely altered the biological activity. So instead of binding to tubulin, these two compounds bind to DNA topoisomerase, and this established the C4 position as a site of biological importance. And so a lot of researchers have also synthesized C4 analogs of podophyllotoxin already, but the reason why we wanted to investigate this further is because there wasn't really a clear trend between the bulk at the C4 position and the compound's cytotoxicity. And so previous group members worked on synthesizing and evaluating these 6 C4 ester analogs.
So once we completed the synthesis of these analogs, we then turned to some biological evaluation. We'll be going a little more in depth about all of these assays later on in the presentation, so here's a quick summary. In our in vitro cell viability assay, we saw that as the bulk on podophyllotoxin increased, or as the red portion of the molecule got bigger, the potency decreased, as you can see in the bottom left corner. Additionally, through flow cytometry assays, we were able to measure the percentage of cells that were arrested at the G2/M phase and saw the same trend of bulk and lowered potency. However, in our ex vitro assay, which is done outside of cells with only our tubulin protein, there was no trend observed between the bulk of the compound and the cytotoxicity. Therefore, we were left with two different questions that we wanted to solve. One was, does increasing hydrophobicity impact cytosolic and membrane distribution? In other words, maybe increasing the bulk on that position doesn't change how the molecule binds to tubulin, but changes how the compound is able to enter the cell. The second question was that maybe there are other non-specific ligand-protein interactions at play. As you all know, the cell has thousands of different proteins, and it's possible that our compounds don't specifically bind to tubulin and may be binding to some other proteins. That could help explain why we saw different results in our in vitro assays as compared to our cell-free experiments. In order to probe these questions further, we then turn to our current work.
So now we are evaluating the biological activity of C4 carbonates and carbamates. So here we significantly increased our analog library. So instead of just 6 compounds, now we have 22 compounds, all of which are novel. And I'll be walking through the synthesis of both the carbonates and carbamates. So for the carbonates, this is a one-step reaction where we add the corresponding chloroformate to podophyllotoxin to achieve the carbonate, and we made 7 carbonates in total. And then as for the carbamates, we had to do some reaction screening in the beginning, or condition screening, and we found that one of the conditions worked, although it was a bit sluggish, but this was a 2-step process. So first, we add the nitrophenyl chloroformate to podophyllotoxin to make the intermediate, and then we add the corresponding amine to make the carbamate, and we made 15 C4 carbamate analogs.
So after we had our library of 22 novel analogs, we once again turn to the biological evaluation. So we'll be walking through a little bit about how all of these assays are run for those of you who may not be familiar. First assay that we ran was an in vitro cytotoxicity assay, which measures the percentage of cells that are still alive in a sample and helps give us a readout on the potency of the compounds. To run this assay, we first, of course, culture our cells and then seed them in a 96-well plate. The number of cells in each well are roughly the same, and so we have a good, stable number to begin our assay. We would then treat this plate with our compound for 96 hours. After treatment, we would add a solution of MTT. In the presence of mitochondrial reductase, the MTT turns into formazin, which is a purple crystal. Therefore, we can quantify the number of cells that are still living based on the number of purple crystals that are present in the well. To do this, we simply dissolve the crystals in DMSO and measure the absorbance through a spectrophotometer. We perform this assay on several different cell lines, many of which are not pictured here, but including colorectal, triple-negative breast cancer, ovarian cancer, murine colorectal cancer, non-small cell lung cancer, and more. Interestingly, we saw that even though these compounds were tested in several different types of cell lines, they all behaved relatively the same, despite the cancers being different. This is analogous to what many people have observed in anti-mitotic drugs, which target the cell cycle. The cell cycle is a ubiquitous process that is present in any cell, and so the activity of the compound does not depend on the cell line it is tested in. Specifically, we found 2 compounds that performed the best throughout this range of cell lines. These were isobutyl carbonate and T-butyl carbonate. Now, some of you may be wondering, how is it that our compounds behave the same across these cell lines? When in this slide, you see that some of the cell lines have greater cell viability as compared to others. Well, the interesting thing about tubulin inhibitors is that they don't actually kill the cancerous cells. As Stella previously mentioned, all they do is inhibit the further growth of these cells. Therefore, different cell lines have different growth timings. For example, one cell line may duplicate in 1 day, while another takes 2 or 3 days. Therefore, we are not able to draw conclusions based on how these compounds perform throughout cell lines, since the growth rate for each cell line is different. In order to combat this, what we did is we normalized all the cell lines and brought them to a baseline. And you can see some of that work pictured here. So as you see, throughout all the different cell lines, the relative trend of these compounds and their concentrations remains the same, as shown with diethyl carbamate. Additionally, with podophyllotoxin, you see once again that it has a steady effect on these different cell lines regardless of what type of cancer it is. So this kind of brings us to the end of our initial in vitro assays. Next, I'll be talking a bit about flow cytometry and some of the basics of it and the mechanics. Now, flow cytometry itself is a fluorescence-based assay, which helps scientists measure different characteristics of cells. In this case, we were interested in looking at the cell cycle and quantifying the number of cells that were in each stage. To do this, the flow cytometer uses a light-based analytical technique, and it is especially important as it is one of the machines in biology that allows you to actually quantify these characteristics. For those of you who may be interested in engineering or mechanics, here's a little bit about how this machine actually works. The flow cytometer has three different parts: fluidics, optics, and electronics. For the fluidics, we inject a sample of cells that are suspended in solution. The most fascinating thing is that the flow cytometer can actually shove all these cells into a single file line and perform a single cell analysis to see what stage of the cell cycle that one individual cell is in. To do that, we have the optics section. In this section, a laser will shoot a beam of light towards the cell, and based on the deflection, the cell can be quantified in terms of relative size, the internal structure, and many other things. Finally, the electronics allow us to take the data from the flow cytometer and create some beautiful graphs that you'll see in the next few slides. So specifically within cell cycle flow cytometry, we're going to be talking a bit about why we chose this assay and how we're able to quantify it. Once again, as Stella mentioned, tubulin is the cell division inhibitor, which means its primary role is in the G2/M phase when the cell is trying to split from 1 into 2. Our compound podophylltoxin works to prevent the splitting of cells. And so for those cells that have been arrested at the G2/M phase by podophylltoxin, they are no longer able to continue throughout the rest of the cell cycle. We're able to quantify the percentage of cells in a sample in each phase through the graphs that we receive from the flow cytometer. As you can see on the right, based on the area under the curve of different sections of the graph, we can quantify what stage of the cell cycle those percentage of cells are in. So we ran all 22 of our compounds through the flow cytometer to get some data on the cell cycle analysis. As expected, podophylltoxin, which is the parent compound and a known tubulin inhibitor, showed the greatest percentage of cells arrested at the G2/M phase, signified by the yellow color. Also, similar to our MTT assays, T-butyl carbonate, which was a lead compound, also showed a good percentage of arrest in the G2/M phase as compared to other compounds. Additionally, also analogous to the MTT assay, the rest of the compounds showed similar percentages in all the phases, which also was analogous to what we saw in terms of the activity in MTT. Now we wanted to take a close look at the different isosteres. As a reminder, carbonates have an oxygen where carbamates have a nitrogen, but in the isosteres, the rest of the substituent is the same. Interestingly enough, in our isobutyl and hexyl isosteres, there was no difference or a limited difference in the percentages of the cells in each of the stages. However, for our lead compound, T-butyl carbonate, there was a significant and almost 2 to 3-fold increase in the percentage of cells that were arrested in the G2/M phase, which was quite fascinating to see.
So for our last in vitro experiment, we did some cell imaging through immunofluorescence staining of beta-tubulin. So first we seeded cells on a 96-well plate and then drugged them with our compounds and then fixed the cells to prevent any further cellular degradation. After that, we added our primary and secondary antibodies. So the primary antibody binds to the beta-tubulin protein, and the secondary antibody binds to the primary antibody, and it fluoresces. So this would allow us to see the beta tubulin proteins in cells. After that, we added DAPI, which is a counterstain, which would allow us to see the nuclei. And then we put the plate underneath a fluorescence microscope to acquire our fixed cell images. And finally, we process the images on a software called ImageJ. And here are the images that we got. So in green, those are all the tubulin proteins, and the blue is the nucleus. There aren't any clear morphological differences between the cells, but as you can see on the left here in the zoomed in pictures, some of the cells are going through mitosis, which is pretty cool.
So after completing these in vitro assays, we then moved on to our ex vitro biochemical tubulin polymerization assay. This assay is a proxy as to how these compounds would work solely in the presence of the tubulin protein, as there are no cells involved. To do this assay, we first have a lot of preparation, which includes freezing down tubulin aliquots, preparing several buffers, and storing our well plate on ice. Tubulin is incredibly sensitive to room temperature and to body heat, and so it must be stored at very cold temperatures until we're ready to run this assay. To run the assay, we first dissolve our compound in a buffer and plate it on the plate on ice. Then we're able to defrost the tubulin aliquots and add a small sample to each of the different wells. Once we have our plate ready with both the drug and the tubulin, we run it on the spectrophotometer and do kinetics for 75 minutes, recording absorbance every 5 minutes. In between these 5-minute intervals, we incubate the plate at 37 degrees to mimic the normal body temperature. After this is done, we're able to normalize and graph the data. Now, there's a lot of things on the slide, but let's break it down. So this assay basically tells us the rate at which tubulin polymerizes. In our case, we want our compounds to inhibit the polymerization of tubulin and prevent it from actually forming these spindle fibers. We have 3 different controls in this assay. One is Taxol, which is shown in red. Taxol is a known tubulin polymerizer, and so it will have a very high activity in this assay. The second is a DMSO control, which is used as a negative control with no compound. And the third is podophyllotoxin, which is shown in yellow. As we've explained, podophyllotoxin is a known tubulin inhibitor. And so oftentimes in our assay, we see it at the baseline with the lowest activity. Now we ran this assay on 3 different concentrations to see whether the inhibition of tubulin was dose-dependent with our compounds. Interestingly, we see that there is still no clear trend between the bulk of the compounds and their ability to inhibit tubulin. Additionally, this data does not exactly correspond with our in vitro data from flow cytometry and MTT, which we also saw in the previous project.
And finally, we conducted computational modeling. So through the software called Schrödinger Maestro, we're able to dock our compounds to the tubulin protein, and it's able to calculate the binding affinity. Similar to what we saw with the ester analogs, there wasn't really a clear trend between the bulk at the C4 position of these carbonate and carbamate analogs and their binding affinities. However, I would like to point out down here in the image below, where I overlaid all 22 compounds on top of each other, all structures are conserved in the same position, except for the substituent at the C4 position.
So now that we completed our in vitro, ex vitro, and in silico experiments, we were able to draw a few conclusions from our work. One of the most important things that we observed was that in silico, the substituent that we added on the C4 position did not change how the compound would bind to the beta tubulin interface. That was incredibly important for us as we began to draw conclusions from our experiments because it showed that regardless of what we would add on the C4 position, the binding affinity would remain relatively the same. Through these assays, we saw that there were similar trends with the cytotoxicity and in our flow cytometry, and we were able to confirm that the primary mode of action is cell cycle arrest through our flow cytometry experiment. Finally, we unfortunately saw that there was no trend observed in our ex vitro biochemical assay. Now, what does this mean? So we started off this project with two main questions, one about cytosolic availability and one about off-target binding. Through our data and our experiments, we're able to conclude that binding is not the primary determinant to potency, and in this case, it is cytosolic availability. Oftentimes in medicinal chemistry, binding is known to be the primary determinant. But in our case, no matter what substituent we added, the relative affinity of binding through both the in silico modeling and our tubulin experiment remained the same. We only saw a true difference in our in vitro experiment where cells were present. This shows that it's perhaps our compounds with greater bulk have more difficulty actually getting across the cell membrane and into the cell where the tubulin is present. So in this case, it is not binding that is determinant, but it's the number of molecules that actually make it into the cell where the tubulin is that determines the potency of that drug. So in all, we're able to conclude that in this case, it is cytosolic availability when it comes to our analogs.
So we're pretty close to wrapping up this project, but some future plans that we have are to fine-tune the MTT assays to calculate the relative IC50 values, as well as quantify intracellular drug concentrations by mass spec. And some lab members are already working on this, but we also plan to synthesize podolinkers to visualize intracellular binding by live cell imaging. And with that, we'd like to thank our advisor, Dr. Edward New, and the rest of our group members for supporting us through this journey, as well as ASDRP and its sponsors for providing the resources and opportunity to conduct this research. And on the bottom right here is the QR code if you would like to see our lab's previous work.
Thank you all so much for joining us today, and we would be happy to take any questions.